So, good evening everybody, and thank you Fabrizio for your introduction. And today I'm going to present my research project in the field of markerless movement analysis by using RGB depth cameras for clinical application. Today the first question of this talk is what is the gait analysis and the movement analysis and why is it so important? In particular, the movement analysis is the study of human motion by using different technologies, and in particular this study has to be systematic and objective. And in particular we can estimate kinematic and dynamic variables, for example joint angles, displacements or forces and momenta. And the importance of these outcomes are that the outcomes can help the clinician to make, for example, an early diagnosis of some degenerative diseases, or to verify the improvement after a pharmaceutical treatment or a surgery. The gold standard in movement analysis is the optical stereophotogrammetry, and this technique consists of putting some markers on the subject on the skin of the subject, and in particular the movements of these markers are acquired by multiple infrared cameras, and of course the results are very accurate. But the disadvantages are the time required for system and subject preparation, and also the cost that amounts to 100,000 euros. So an alternative is the markerless movement analysis that doesn't require markers on the skin of the subject. And so, of course, the experimental sessions are faster and simpler with respect to marker-based system. And in this case, there are different research topics, and in particular, some of them use multi-camera approaches, or some uses single camera approach. And for the algorithm point of view, it's possible to choose a machine learning approach or some deterministic approach, for example, model-based. And so in this case, my research activity is focused on the markerless movement analysis by using a single RGB depth camera. So first of all, an RGB depth camera is a camera that includes two types of sensor. An RGB sensor that produces a normal color image, and a depth sensor that produces a depth image, so an image that shows the distance of all objects in the field of view of the camera. And so I use this camera to for example, do markerless gait analysis on subjects affected by cerebral palsy. And so the aim of this study, of my study, is to propose and validate a portable and low-cost markerless system by using a single RGB depth camera for estimating, for example, kinematic angles or some spatiotemporal parameters for follow-up screening purposes in subjects affected by cerebral palsy. So this is the entire flowchart of my process, in which the first step is the gait cycle segmentation. This step aims to identify the central gait cycle in order to calculate some spatiotemporal parameters, for example, the stride length or the step length or the gait speed of the subject. So in this case, the identification of the foot contact with the ground is not simple, because in this case there are some patients who don't impact to the ground with the heel as in a normal gait, but with the toe or with the flat foot. So it's possible to segment the feet of the subject, because as you can see, the subjects are asked to wear two different color socks to distinguish between the left and right foot by using a simple color filter. So, for each of segmented foot, it's possible to identify the heel and toe of this image, and for each of these segmented foot, it's possible, it's possible to extract both coordinates, so vertical and anteroposterior coordinates of both heel and toe. So between heel and toe, the point of contact with the ground is the extremity that first reach the stationary condition of both coordinates, so vertical and anterior-posterior coordinates. And in particular, in this case, when the subject impacts in this way, the heel is the point of contact because it reaches the stationary condition before the toe. So after the identification of the gait cycle, it's possible to segment, so to extract the subject from the image. And in particular, for this purpose, an automatic segmentation algorithm was designed and applied on RGB image, so on color image. And this algorithm is based on the analysis of the images to find a threshold that separates the subject from the background. So after the identification of the subject, it's possible to use a model to estimate the kinematics and the kinematic angle of the subject. And in particular, my model is composed by 3 segments: the foot, shank and thigh, and 3 anatomical landmarks, so lateral malleolus, lateral epicondyle, and retrocanter. So the point of this step is that to create and calibrate this model, the clinician is asked to manually identify on a static image the point of interest, so these 3 anatomical landmarks. And after the identification of this model, it's possible to track all these points in all frames of the gait cycle. And in particular, I use a bottom-up approach tracking, starting from the foot and to arrive to the identification for, of course, each frame of the gait cycle. And in particular, it's possible to identify the foot, as I said before, by using a color filter. And so, after the identification of the foot in each frame, we have to estimate the position of lateral malleolus in this frame. So, to achieve the same, I align the foot model to the foot in each frame by using an iterative closest point. This algorithm is an optimization technique to find the rototransaction matrix that minimizes the distance between the foot model and the segmented foot. So by multiplying the rototransaction matrix to the position of lateral malleolus in the model, it's possible to estimate the position of this anatomical landmark in each frame. So, starting from the position of this lateral malleolus, it's possible to draw this circle to identify this region. And so, in this case, the problem is that the 2 legs are overlapped. And so, for example, during the swing motion of the leg, it's possible to notice this situation. And so, to extract the foreground shank, we had to use the information provided by the Dept image. And for example, in this case, I computed the histogram of Dept value inside this same cycle. So, by analyzing this histogram, it's possible to notice 2 distributions. The first one represents the foreground shank, and the second one represents the contralateral one. And of course, it's possible to think that the foreground shank is characterized by the lowest depth value since it's closer to the camera. And so, in this way, we can extract the foreground shank. So, as I said before, for the foot, it's possible to estimate the position of the lateral epicondyle, so the knee, by using the iterative closest point between the shank model and the shank previously obtained. So, the same procedure is applied for the thigh to obtain the position of retrocanter. So once we can extract all the anatomical landmarks, it's possible to calculate all these joint angles, and it's possible to obtain this curve. For example, in this case, I reported the curve of the knee of this subject. And to validate this procedure, I compare the markerless outputs with the outputs of marker-based system, that is the gold standard, as I said in the introduction. And in particular, we acquired 11 children with bilateral cerebral palsy, and I used the Kinect 2, a homogeneous background, and two ample sorts of different color, and two LED lamps to improve the light condition. And of course, I used the marker-based system for the validation, and for each subject I acquired 6 dynamic trials, 3 left and 3 right, and 2 static trials, 1 left and 1 right. For the validation, for the spatiotemporal parameters, I calculated the absolute error in percentage between markerless and marker-based system, and I obtained this interesting error because they are encouraging. And for what concerns the joint angle, I calculated the root mean square error between the markerless curve and the marker-based curve in order to obtain this error that are due mainly for different definition between the axes of the two systems. And in particular, we can notice that the angle shows the highest error because the Kinect 2 provides these blurred images due to the automatic exposure time of the camera. This markerless system is suitable for clinical gait analysis because the results are very encouraging. And of course, the advantage of this model-based technique with compared to machine learning approach is that we can give the possibility to clinicians to define and calibrate the model to calculate some parameters of clinical interest. And for future developments, we are working with the new version of Kinect 2, the Azure Kinect, to avoid blurred images that are causing the error in Kinect 2. And we are working to obtain the 3D gait analysis. So the second activity of my project is the markerless upper limb motion in infants. So the problem is in infants is that deviation from the typical development have to be detected as early as possible to leverage the neuroplasticity of the brain in early infants. So the clinical need is the estimation of quantitative parameters to be used as biomarkers predictive of movement disorders. So the aim of this work is, as the previous project, use a single RGB depth camera for estimate the kinematic of upper limb motion, and this system is designed to be used at patients' houses. And this is the experimental setup. So a baby was sitting in a chair, is placed in front of the camera, that in this case is the Intelligent Sense D45, so it's different from the previous. And while in the previous application I used a model-based approach, in this case we used a deep learning approach by using the DeepLab cat algorithm that estimates all these anatomical landmarks, so left and right shoulder, left and right elbows, and left and right wrist, and also the relevant confidence level that provides an information of the reliability of the estimates. So, knowing the pixel location of each point, it's possible to extract the depth value for each frame. We can notice that in an original depth there are some artifacts. So the first artifacts are the zeros due to the presence of this black area. So during the movement a marker could fall on one of these black points. And the second and the most important problem is the occlusion of, for example, in this case, the left shoulder is covered by the head of the patient. So to solve this problem, it's possible to use the information of the confidence level, because I set a threshold on the confidence level to remove the frames below this threshold. So all gaps are filled by applying a span interpolation to obtain the 3D coordinates estimation. So to preliminary validate this method, we acquired two cases of a doll on a turntable. So the turntable is placed parallel to the camera, and in the second acquisition, the camera was tilted at 45 degrees to facilitate the observation of the occlusion. So to test this method in presence of the occlusions. So I calculate the mean absolute error of these quantities, so some anthropometric and kinematic quantities, and as expected, of course, the performance at 0 degree are better than 45 degree, because in the latter there are some occlusions, so some problem. And to conclude, the future development of this work is to validate the method on real baby to try to estimate parameters for to be used as biomarkers. So this is my research group, and thank you for your attention.